Hello viewers, welcome to our channel Biological Concepts. In today's video, we will discuss some questions on mutations. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question, a physical or chemical agent able to cause mutation in a DNA molecule is known as option 1 mutant, option 2 auxotrope, option 3 mutagen, option 4 mutated gene. And the correct answer is option 3, that is mutagen. Now let's discuss about mutagen. In genetics, a mutagen is a physical or chemical agent that permanently changes the genetic material. This genetic material is usually DNA, and it increases the frequency of mutation above the natural background level. This mutagen may be either physical mutagen or it may be chemical mutagen. The physical mutagen include the UV ionizing radiation. The ionizing radiations are X-ray, gamma ray, etc. And the examples of chemical mutation include hydroxylamine, nitrous oxide, reactive oxygen species, alkylating agent. Although the mutagen cause the mutation in DNA molecule, but not all mutations are caused by mutagens. There is so-called spontaneous mutation, which occur due to spontaneous hydrolysis, error in DNA replication, and repair and during recombination. So these spontaneous mutations are not caused by the mutagen. These are occurred due to spontaneous hydrolysis, error in DNA replication, repair or recombination. That is, this spontaneous mutation occurs spontaneously, not by the influence of the mutations. Next question. A mutation that results from a single nucleotide alteration are known as option 1 polar mutation, option 2 point mutation, option 3 back mutation, option 4 leaky mutation, and the correct answer is option 2 point mutation. A point mutation is a genetic mutation where a single nucleotide base is changed, inserted or deleted from a DNA or RNA sequence of an organism's genome. This point mutation usually takes place during DNA replication. This point mutation may be either missense mutation or silent mutation or nonsense mutation when a single base is changed, or it may be insertion or deletion mutation, that is frameshift mutation. We will discuss about this different kinds of point mutation shortly. Next one is polar mutation. From the name polar, this polar mutation affects the expression of downstream gene. Next is back mutation. In back mutation occur of an existing mutant gene. It restores to its previous form. So when back mutation occur in an existing mutant gene, it restores it to its previous form. As a result, the mutated gene is restored to the original nucleotide. Next is leaky mutation. A mutation that does not cause a complete loss of function of a wild type phenotype is known as leaky mutation. Next question. A mutation that results continuous expression of a gene known as option 1 polar mutation, option 2 constitutive mutation, option 3 back mutation, option 4 is leaky mutation. And the correct answer is option 2, that is constitutive mutation. A mutation that results continuous expression of a gene or a set of genes that is normally subject to regulatory control. Due to this regulatory control, these genes may be either repressed or expressed. Okay, but in constitutive mutation, this gene, there will be no regulatory control and there will be continuous expression of the gene or set of genes. A mutation that results in an increased constitutive synthesis by a bacterium of several functionally related inducible enzymes. Such a mutation either modifies an operated gene so that the repressor cannot combine with it, or modifies a regulated gene so that repressor is not formed. Due to the modification, when the repressor is unable to bind to the operated gene, as a result, there is no repressors of this gene, so there is continuous expression. Other hand, when the repressor is not produced, there is a repression of this gene. As a result, there is the continuous expression of the gene, and this causes constitutive mutation. Next question. A mutation in which there is deletion of one or few nucleotides are known as option 1 frameshift mutation, option 2 silent mutation, option 3 nonsense mutation, and option 4 none of the above. And the correct answer is option 1, that is frameshift mutation. A frameshift mutation, also called framing error or reading frame shift, is the genetic mutation that is caused by insertion or deletion of a number of nucleotides in the DNA sequence that is not divisible by 3. Due to the triplet nature of gene expression by codons, the insertion or deletion can change the reading frame, the grouping of the codons, resulting in a completely different translation from the original. The earlier in the sequence of the deletion or insertion occurred, the more altered the protein. Why there is the different result in a completely different translation form of the original? Because suppose if this is a base sequence A, T, C, G, A, C. Okay. Suppose these two are deleted. Okay. It is this 3, then this 3, then so on. Okay. If these two are deleted, then the sequence will be like T, A, C, G, A, C. So there will be another different type of protein product. Okay. This results in the change in the entire reading frame. This is TAC, CAG, AC something, ACG or ACA. So here the deletion occurred after the TAC, CA. So TAC is remain same. Then if this CAG is replaced, totally replaced CAG, and ACA or GRC replaced, and it causes GAC, another one. Okay. Next question. Induction of mutation induced by X-ray was discovered by option 1 Muller, option 2 Morgan, option 3 Hugo de Veris, option 4 is Weizmann. And the correct answer is option 1 Muller. Hermann Muller studied the hereditary characteristics of fruit flies in 1927, and he has discovered that number of genetic mutation observed in fruit flies increased when they are exposed to X-ray. Next question. Sickle cell anemia is a classical example of option 1 frameshift mutation, option 2 point mutation, option 3 nonsense mutation, and option 4 is none of the above. And the correct answer is option 2 point mutation. Although these two, that is frameshift mutation and nonsense mutation, are also the point mutation, but here these two options will not be, and the answer will be point mutation, because the sickle cell anemia is a classical example of missense mutation, and missense mutation is one kind of point mutation. As in this option missense mutation is not present, only point mutation is present, and frameshift mutation, nonsense mutation present. So the answer will be point mutation, because it is neither frameshift mutation nor nonsense mutation. So the answer will be point mutation. So sickle cell anemia is caused by substitution of a single nucleotide in the beta globin gene sequence, which eventually lead to the replacement of the amino acid glutamic acid with valine, affecting the final product. So in sickle cell anemia, the glutamic acid is replaced with valine. So this one is the missense mutation, that is a point mutation. Next question. Mutation induced by 5' uracils is option 1 transition mutation, option 2 frameshift mutation, option 3 transversion mutation, and option 4 is backward mutation. And the correct answer is option 2, that is transition mutation. Sorry, this will be option 1. A transition mutation is a point mutation that changes the purine nucleotide to another purine. The purine are adenine and guanine. So this adenine will be either replaced with G, or guanine will be replaced with A, or the pyrimidine nucleotide to another pyrimidine. That is, pyrimidine are cytosine and thymine. So this cytosine will be either replaced with thymine, or thymine will be replaced with cytosine. Whereas the transversion is the replacement of purine with pyrimidine or vice versa. Okay, these transition mutations are 10 times more common than the transversion. The 5-bromouracil is a base analog of thymine. So 5-bromosyl is the base analog thymine which can be incorporated into DNA. It is a well-known mutagen causing transition mutation by mispairing with guanine rather than pairing with adenine during the replication. So this is one kind of induced the frameshift transition mutation. Next question. Most striking example of frameshift mutation is found in option 1 clotted blindness, option 2 thalassemia, option 3 sickle cell anemia, and option 4 is all of the above. And the correct answer is option 2 thalassemia. Thalassemia is an example of deletion mutation in hemoglobin gene. As we have already discussed that deletion mutation and insertion mutation is one kind of frameshift mutation. So this thalassemia is a example of frameshift mutation. Thalassemia is a group of blood disorder which are caused by mutation in the HBB and HBA genes present on the chromosome number 11 and chromosome number 16 respectively. The unequal crossover within the globin gene complexes causes deletion, which in turn leads to the backward shifting of the reading frame. As it causes deletion, and this mutation is known as deletion mutation. So this deletion mutation is one kind of frameshift mutation. Another example of frameshift mutation include the Crohn's disease, TSH disease, cystic fibrosis, etc. The specific frameshift mutation in CCR5 gene has also been linked to HIV resistance. Next question. Transition type mutation occur when option 1 AT is replaced with GC, option 2 AT is replaced with CG, option 3 AT is replaced with TA, option 4 CG is replaced with GC. Here A means adenine, T means thymine, G means guanine, and C means cytosine. So answer will be option 1, that is AT is replaced with GC. We have already discussed that transition mutation is a mutation that changes purine nucleotide to another purine, that is A to G or G to A, or a pyrimidine nucleotide to another pyrimidine, that is C to T or T to C. Then it is called transition mutation. Approximately 2 out of 3 single nucleotide polymorphisms are transition. Transmission can be caused by oxidative deamination and tautomerization. Transition appear more often in genomes, possibly due to molecular mechanism that generate them. 5-methyl cytosine is more prone to transition than unmethylated cytosine due to spontaneous deamination. This mechanism is spontaneous important because it dictates the rarity of CpG island. Next question. A mutation that does not alter the protein product is known as option 1 nonsense mutation, option 2 missense mutation, option 3 transversion mutation, option 4 is silent mutation, and the correct answer is option 4, that is silent mutation. Silent mutations are mutations in DNA that do not have an observable effect on the organism's phenotype. Silent mutations are a base substitution that results in no change in the amino acid or amino acid functionality when the altered messenger RNA is translated. For example, if the codon AAA is altered to become AAG, the same amino acid lysine will be incorporated into the peptide chain because both AAA and AAG codes for the lysines. As a result, there will be no changed phenotypic expression of the gene. Next question. Original DNA segment is ATGCGC. The mutated DNA segment is ATTGCGC. Which type of mutation occurred here? Option 1 deletion mutation, option 2 insertion mutation, option 3 is transversion mutation, option 4 is backward mutation. So in In backward mutation, we have already discussed the original nucleotide will be mutant nucleotide will be changed to the original nucleotide. Here it does not occur here, so it is not backward mutation. So in the original DNA molecule there is 1, 2, 3, 4, 5, 6 nucleotide. There is 6 nucleotide, and here is 1, 2, 3, 4, 5, 6, 7 7 nucleotide. So in original DNA molecule there is 6 nucleotide, and in mutated DNA molecule there is 7 nucleotide. So there is insertion of 1 nucleotide. So if there is insertion of 1 nucleotide, then it will be called insertion mutation. So the option will be insertion mutation. So in insertion mutation is the addition of 1 or more nucleotide base pairs into DNA sequences. Here insertion of 1 nucleotide occur here, because in the original DNA molecule there is 6 nucleotide bases, whereas in the mutated DNA sequence there is 7 nucleotide bases. So it is insertion mutation. Next question. The original DNA segment is ATGCGCTT. The mutated DNA molecule segment is ATGCTT. Which type of mutation occur here? Option 1 deletion mutation, option 2 insertion mutation, option 3 transversion mutation, option 4 is backward mutation. So in the original DNA molecule there is 1, 2, 3, 4, 5, 6, 7, 8 nucleotide. Whereas in the mutated DNA segment there is 1, 2, 3, 4, 5, 6 nucleotides. So in the mutated DNA molecule there is deletion of 2 nucleotide bases. So this mutation will be deletion mutation, and the answer will be option 1, that is deletion mutation. In deletion mutation we have already discussed that there is deletion of 1 or more nucleotides from the DNA molecule. Okay. Next question. Proflavin or acridine orange induces which kind of mutation? Option 1 frameshift mutation, option 2 missense mutation, option 3 is transversion mutation, option 4 is nonsense mutation, and the correct answer is option 1 frameshift mutation. Proflavin and acridine orange are intercalating agent. They intercalate between the bases and causes the frameshift mutation. We have already defined what is frameshift mutation. Okay, just check it. Muller was first to produce induced mutation in dash by exposing them in X-rays. Option 1 Arabidopsis, option 2 Genopas, option 3 E. coli, option 4 is Drosophila, and the correct answer is option 4 Drosophila. Muller in 1927 discovered that the number of genetic mutation observed in fruit flies, that is Drosophila, increased when they are exposed to X-ray, that is the ionizing radiation. The action of UV rays on DNA to induce mutation is the option 1 formation of thymine dimer, option 2 methylation of base pair, option 3 deletion of base pair, and option 4 is alkylation of base pair. And the correct answer is option 1, that is formation of thymine dimer. The major role in inducing sunlight dependent skin cancer in mutation is assigned to the cyclobutane pyrimidine dimers. Next question. The original DNA segment here is ATGCGA and mutated DNA molecule is ATGTGA. Which type of mutation occurred here? Option 1 missense mutation, option 2 nonsense mutation, option 3 silent mutation, option 4 is backward mutation. Here in the original DNA segment, that is ATGCGA, and in the mutated sequence it is ATGTGA. So the here 1, 2, 3, 4, this fourth number of nucleotide base is changed, that is cytosine is replaced with thymine, and so there is single base change. So it may be either missense mutation, or it may be nonsense mutation, or it may be silent mutation, and the answer will be option 2 nonsense mutation. Why it is nonsense mutation we will discuss shortly. In nonsense mutation is a point mutation in a DNA sequence that results in a nonsense codon or a premature stop codon in the transcript RNA and leads to truncated, incomplete, and possibly non-functional protein product. So when this original DNA molecule is changed to this mutated segment, it results in ATG TGA. This is the segment. So in the mRNA, it will be UGA. Okay, UGA is a stop codon. We know that UAA, just remember UAA, UAG and UGA. These three are stop codon. In the mutated DNA segment, it is mutated to ATG TGA. The mRNA will be the UGA segment when ATG UGA. So UGA is the stop codon. So there is stop, which results in nonsense codon. We know that the mutation where when the mutation that results in the formation of nonsense codon, then it known as the nonsense mutation. Here it generates a nonsense codon, so it that is a premature stop codon in the transcribed mRNA. So it is a nonsense mutation. Nonsense mutation are not always harmful. The functional effect of a nonsense mutation depends on the many aspects such as the location of the stop codon within the coding gene. For example, the effect of nonsense mutation depends on the proximity of the nonsense mutation to the original stop codon, the degree to which the functional subdomains of the protein are affected. As nonsense mutation leads to premature termination of the polypeptide change, they are also known as termination mutation. Here, the CGT causing guanine to be replaced with TGA in the DNA sequence, which results at the protein level the replacement of arginine to stop codon by the stop codon. So CGT produce the arginine, whereas TGA, when it is replaced with TGA in the mRNA sequence, it will generate the UGA, and this UGA is a stop codon. So it is replaced by stop codon, and this is nonsense mutation. That's why it is nonsense mutation. Okay. Next one, the original DNA segment is AAC-CGT. The mutated DNA molecule segment is AAC-CTT. Which type of mutation occur here? Here also there is mutation in one nucleotide base. So this is also a point mutation. Here options are missense mutation, nonsense mutation, silent mutation. And 4 is backward mutation. And the answer will be option 1, that is missense mutation. In missense mutation differ from nonsense mutation since they are point mutation that exhibit the single nucleotide change to cause substitution of a different amino acid. In the DNA sequence, CGT causing guanine to be replaced with CTT in the DNA sequence. This results in the protein level replacement of arginine by leucine. Okay. So if as this arginine is replaced with leucine, so it is missense mutation. Let's discuss about another examples of missense mutation. In the 5' to 3' DNA sequence, this is the 5' to 3', this one is 3' to 5', and look at this, the same nucleotide bases in the 5' to 3' position, only the T is replaced with U. Okay. So remember that both the 5' to 3' DNA sequence and 5' to 3' mRNA sequence will be same, except the thymine will be replaced with uracil in mRNA. Okay. And when this G is replaced with A, it results in the protein product aspergine. This serine is replaced with aspergine. So this is missense mutation. Okay. If you like this video, please like, share and subscribe to this channel. Thank you.